It's really my pleasure to be here today and to speak to all of you. I was speaking to Dave right before this, and we were commenting on the fact that there are so many patients here today. Dave said around 29. And one of the issues that we feel like faces Castleman disease is there's not as much awareness around this disease as really there should be, given the prevalence of the disease. So we're excited that with events like this, all of you can feel like you're having a family, other people who have gone through the same thing you can talk to about this. It warms my heart to see everybody here. So with that, I'll get started. As Dave mentioned, my name is Jason, and I'm the Chief Scientific Officer for the Castleman Disease Collaborative Network. And I also do research at a place called the Broad Institute of Harvard and MIT up in Boston. So I wanted to start off the talk. I know it's right after lunch, everyone's a little tired because you got that food coma you're about to go into. So I wanted to make it a little more interactive, so I thought we could start. Could anyone tell me what desert this photo is taken in? Anyone? Want to guess? Sahara. Okay, I hear Sahara, Yogi. A few guesses. All right, so what was that one? In Utah. Not exactly close to Utah. You'll see where I'm going with this, though. So here's a satellite map. Good guesses, though. Really good guesses. So here's a satellite map of where the picture was taken from. You can see there's some names of some features nearby. So still, getting closer. Someone said the moon. All right. So we're getting a little closer there. All right. Think even a little further away. Mars. Right. Mars. So if we go to a selfie of the photographer who took the first photo, this is the Curiosity rover that NASA sent to Mars in 2012. It's absolutely amazing, right? We sent a probe to Mars to do research on a planet super, super far away, this mysterious thing that we've looked at for a very long time. It's been very hard to understand. And it wasn't until we actually sent this robot, about the size of a car, to a different planet that we could actually start to answer some of the questions we've had about Mars for really millennia. That little thing right there, that's a hole that the rover drilled into the surface of Mars just to ask a question about what minerals and what atoms make up the Martian soil. Some of the photos that the rover has taken have provided evidence that Mars used to have giant bodies of water on it. And now people think that within 100 years we could have colonies of humans living on Mars, large part because of all the research we're doing right now. These missions we're sending out, they're giving us all this new important information. And really, this isn't going to be the last Mars rover we send. We're going to send another one in 2020. We're going to send another one after that because we want to keep answering more and more of these questions until we can get the answers we want to know. And really, medical research is kind of the same way. So if we're here on Earth and we want to do research on Mars, we can have the best people in the world getting together saying, we want to study this thing that's mysterious and far away. It might be hard to do, but we're confident that we can start to do this. So if we think about other areas of medical research that over the past few decades have seen enormous progress, we can think about a cure in hepatitis C coming out in the last few years. We think about amazing new treatments in cancer for many of the cancer patients. That's something we're still fighting, but all the research we're doing is really pushing that ahead. And we're actually in the middle of a biomedical renaissance right now, where because of all the progress we're making for all these different diseases, we have a lot of really exciting tools that we can use to ask just about any question we want. The problem is, we're over here on Earth, and we have to get our probe to Mars. The researchers are stuck on Earth. When we sent the probe to Mars in real life, we had NASA to send our probe to Mars. But the problem is researchers don't know how to make space shuttles. They can't actually get to Mars themselves. So how do we send a mission to our Mars? How do we send that to Castleman disease? Who is NASA for us? And really one of the reasons I'm so excited to see so many of you here today is that patients are really like NASA for Castleman disease. You guys are the bridge between the researchers and the disease, the way we start to get probes out to send those missions, those research projects Dave was talking about earlier that I'll talk about soon. This is really the way we ask those questions. And I'll get into some details here. So I think it's a good analogy, right? The Accelerate Registry that Dave was talking about earlier, this is creating a map. Where do we want to send that rover? Where do we want to send that next research project we're going to do? How do we prioritize that? We have to know more about the way patients with the disease normally, the disease progresses. So the registry is going to tell us that. And then also, when we want to ask a question, when we know where to send the probe, we have to have the probe to send there, so we have to have the tissues that we can actually use to ask that probing question. So the Castleman Biobank is also a key part here in doing research on Castleman disease. So really, just as NASA is going to keep sending many more of these probes to Mars to keep asking additional questions, as a community together, we're going to keep sending out more and more research projects until we get to the point where we understand this disease, we can develop new treatments, and then everyone has a sufficient treatment for Castleman disease. Important to emphasize, not just the patients here today, those certainly in the online community as well. The more patients we can get involved, the more individuals that contribute samples, the more of these missions we can send. And each mission is critical because it allows us to answer another question. And in research, that's what it's all about, right? We want to ask these important, big questions. What is Castleman disease? What are the molecules and the cells that drive Castleman disease in patients that have the disease? So the way we answer these questions are with research projects. I'll get into those in a little bit. But first, I just want to emphasize, we're really confident that with all the tools that have been developed for the other diseases, we can start to take those in, use those for Castleman disease, understand the biology better, and find better treatments for the disease. Why am I so confident in this? We've done it before. Think about it. Every time you go to the hospital, everything you get treated for, sometime in the past, that treatment didn't exist. Someone had to discover that, right? We're exactly at that point right now for Castleman disease. And as Dave said earlier, it is this community who's going to bring together the patient samples, the expertise, the know-how, everything we need to make those advances, to understand the biology and drive the new treatments. So getting into how this really works, when patients, researchers, and physicians get together, we start building out this research infrastructure, the biobank and the registry. This provides a basis for researchers not just in Philly, but around the world to do Castleman disease research. So these research projects allow us to answer these new questions and nominate new treatments that you can then test in a clinical trial. And then, of course, in a clinical trial, you can test, does it really have an effect on patients? The FDA can then approve the effective therapies, just like siltuximab was recently approved. We're going to look to get additional ones done in the future, of course. And that leads to new treatments for patients. So this is really how the research cycle works. It's key to point out here as well, not only are the patient samples and patient participation absolutely crucial to this, but also financially, this takes a bit of support as well. So the Castleman Disease Collaborative Network, all of the people who are out there being warriors for us, the fundraisers that we hold as an organization, all of our donors, everyone is helping drive these projects forward, driving us towards research that will lead to new treatments for patients. So this is how we're going to get it done, coming together as a community. So I wanted to go over some of those research projects so you can understand when you see all the names we have for our research projects, Hunt One, Speed, Fast, kind of what all this means. We really like the acronyms for our research titles. So thinking broadly, we have a few different types of research projects we're doing, and we like to put those under the type of question they're trying to answer for us. So we want to know what causes Castleman disease, and we have projects in genetics and one in virus hunting. Dave mentioned we actually had the answer to this one already. We also want to know what molecules and cells drive Castleman disease, and we have several studies to look at the molecular drivers and also the cellular drivers of Castleman disease. So now I'm going to go through some of the details of some of these projects so you get a pretty good idea on where the money is going, where the samples are going, and the types of answers we're looking to get. So starting off, the Castleman Genome Project. This is a big genetics project. The goal here is to discover and basically look for changes in the DNA of patients with Castleman disease, where we're going to read 20,000 genes in every patient that comes in, and we're going to look for spelling errors in each of those genes to see, if there's a spelling error in enough patients with Castleman disease, maybe that's what's causing the disease. So we need a lot of patient samples for this. Right now, this project is actually underway. It's funded. It was $63,000. And we took in patient samples from younger Castleman disease patients, some of their family members. We're excited that within the next few months, we should start to see data from this study. It's going to be a very powerful study for us. We're really excited about it. There's the somatic mutation search, which is another genetics project. And this is a little different. So the first one, we're looking for genetic differences in your DNA when you're born. And here we're looking for genetic differences that can arise in your DNA while you're alive. Just your cells are dividing, and all of a sudden one day they make a mistake when they're writing down all the DNA. And it's that mistake that can then lead to a somatic mutation, which may be something that drives Castleman disease. We don't know. We have to do the study to find out. So to do this, as I mentioned, we'll look for these newly developed mutations in 300 genes that we're going to sequence really deep. So we're going to read the same gene many, many times just to be very confident, because sometimes those can hide in there. And this study is going to cost around $40,000. We've actually got the funding for this already, but Dave pointed this out earlier. We need a lot of samples for these studies. So here, the main obstacle really is collecting the final samples we need for this. And we're going to be isolating DNA from these samples, looking at adults with Castleman disease, and trying to answer this question: are there somatic mutations driving Castleman disease? We have a virus hunting study. Dave mentioned we actually have the answer to this one. Here we're using a technology that was previously used to search for the West Nile virus. We used that recently to look for a new virus in Castleman disease. And as Dave mentioned, this study cost about $57,000. We had collaborators from Norway, Japan, all over the US to get this study done. It was very challenging. But in the end, we came up with a very important answer, as Dave mentioned earlier, which is that there was no new virus in these samples. So that tells us, Dave had that list of hypotheses about what could cause the disease. It allows us to tick one of those off the box. And now we can go back to these other studies I mentioned, the somatic and germline genetic studies, to start to ask questions a little deeper there. So moving on, of course, the sequel to the Hunt1 study has got to be Hunt2. And this is actually a study to look at molecular drivers of the disease. So here we're looking at molecules called RNA in each of the cells in the lymph nodes of patients. And we hope that by doing this, we can look for differences in those RNA molecules between the Castleman patients and healthy individuals, or individuals with related diseases that are not Castleman disease, to try to start to tell the difference, what's specific to Castleman disease and what's just a normal RNA that you would expect to be there anyway. So then these RNAs, we start to find they can become, they can tell us about pathways that are turned on, they can tell us about the cells, which we'll get to in a little bit. But they're a key clue in the story about what's happening in patients with Castleman disease. So this is actually a study where we're still looking for funding here. We have the samples available for this because we're using the same samples we used for Hunt1. Once again, this is frozen samples from collaborators from as far as Norway and Japan. Really exciting we're able to do this study. Only by coming together with all the patients, all the physicians from around the world can we get this done. Moving ahead, some additional molecular driver studies are SPEED1 and SPEED2. So both of these studies are basically asking the same type of question, but they're asking it on a different scale. So both of these questions are studying inflammatory molecules, sometimes called cytokines or chemokines, that are in your blood. And these are the molecules that can make your immune system get really activated. And we're looking at over 1,100 of these in patients. The proteomics approaches we're using have also been used in other diseases like diabetes to try to figure out, is this patient going to get diabetes in the future? So those are some of the other ways this technology is being used, and we're using it for Castleman disease to better understand it here. Now, SPEED1 costs around $23,000. This takes serum from 9 patients, actually from UAMS. We're working with Dr. Van Ree on this study. and looking at the patient data to really figure out what's going on in these patients. And we were so happy with the early results from this study that we decided to also write a sequel to Speed 1. And of course, everyone knows what the sequel to Speed 1 is. It's Speed 2, which was a 1997 film starring Sandra Bullock. But seriously, we did a second study called Speed 2. It's still the same inflammatory molecule study. But actually, as I said, it's much bigger this time. We were excited about the results the first time through, so we scaled it up. This time it's $350,000, but we're looking at 240 Castleman disease patients and actually over 100 different control samples from related diseases. This is going to give us a very precise understanding of what's going on in Castleman disease, and it'll allow us to think about, well, here's the pathway that's turned on, here's something that we can potentially target with one of these therapies. So right now, we're fortunate to say that this study is funded, and we are finishing up collecting samples from collaborators around the world. You can see we're going across a few different continents here, and we're really excited about this study. Should be getting results probably within the next year. So moving on now to one of our cellular studies. So a cellular study, we're looking at the individual types of cells in the bloodstream or in the lymph nodes and kind of asking them, are you the same cells we've seen in all the other patients? Are you a little bit different? So we can use a technology to ask that question called flow cytometry, where you look at 50,000 cells from every single patient. And you can ask every single cell, 10 different questions. Are you CD4 positive? Yes or no? Are you CD8 positive? Yes or no? And I know that sounds like gibberish, but trust me, it's very important to a scientist whether a cell is CD4 or CD8 positive. So we can ask these questions, and we can start to understand what are the very small differences between the Castleman disease patients and healthy patients or patients with related diseases. And once again, that gives us what's specific about Castleman disease, what can we go after therapeutically. So this study will cost around $20,000, and we're continuing to collect samples for this study. So this is one of the areas actually that when we collect blood, for example, for today, some of these samples may be used towards this study. So, I've gone over some of the studies we're doing right now. I wanted to also just throw out there some ideas for studies we're thinking about in the future, just briefly. These aren't things we've really started yet, but this is where we see the future of this research going. So first, there's an exciting technology called MIBI, which is multiplexed ion beam imaging. I know it sounds very fancy. We're going to give it a name like FASTER or something, so it's going to be good. It's going to be like the fastest study, something like that. And after we give it a name, then we can share with the details. But this is a study, for example, where with about $100,000, we could look at 100 different samples from lymph nodes of patients. And the same thing I was saying earlier, where you can look at, ask yes or no questions to 50,000 different cells. You could do that, but you could ask even more questions. And instead of asking it in cells in the blood, you could do it on a solid piece of tissue in a lymph node. So you could not only say, well, yes or no, are you CD4 positive? But you could also say, oh, you're in the middle of the lymph node. That means something really different than if you're on the outside of the lymph node. So that gives us additional information we can use to start figuring out what are the cells and molecules driving this disease. So I've been talking a long time about the cells and the molecules and the DNA and everything. And you might wonder, how does any of this actually lead to a therapy, right? If you're just asking lots of questions, yes or no questions, what do you do with that? So really, the future of Castleman disease research isn't just these exciting discovery studies, it's also functional studies. So what that means is, as an organization with the other scientists and researchers who are doing this around the world, we're going to figure out ways to model Castleman disease in a petri dish or in an animal, so we can start to ask, yes or no, does this drug affect the phenotype of Castleman disease in those models? And when we can start to ask questions like that, suddenly we have a very powerful tool to ask questions about what drugs we can actually use and start to move into patients. So that's the point at which all of these studies start to become something we can really turn into questions about, is this a therapy we can start thinking about bringing to patients? And this is going to happen. We already have been talking to people. We were speaking with some folks up at Harvard a few weeks ago. They had an idea on a way we could do this. This really hadn't been done before, but because of the studies we're doing now, the SPEED 2, the HUNT 2 study, the Castleman Genome Project, somatic mutation, all of the tissue donations, we can do these studies within a year or two. We understand what we need to do. We just need to get the results from the studies. Then we'll be able to set this up, ask these questions about additional therapies. And this is where new therapies for the disease can come from. So that's kind of an overview of the research that's going on right now and where we see research going in the near term. Now, to think about themes for research in the future, of course, we're going to build out the infrastructure to empower other researchers. So the Accelerate Patient Registry and the Castleman Tissue Bank, as I mentioned earlier, are going to be key, not just for the patients here, but for all the patients online to participate in and contribute to, so that we can really ask these research questions, send out our missions to Mars, to the planet Castleman Disease, and ask those questions with our probes. We're already doing this through the patient registry and biobank. We need more people in those. We're doing these hypothesis-generating studies, the one I had listed at the top earlier, genetics, molecular, cellular drivers. We'll continue to do the best hypothesis-generating studies we can. As I said, we'll also start to test the hypotheses with functional studies where we can start to look at drugs. Finally, when we start to get a good idea of the type of target we want to go after, we have our functional studies, everything looks good, we can start to think then about the clinical trials we would build out around these new drugs. So this is the path forward for Castleman disease. This is how it's going to happen for everyone in this room, everyone around the world. And just think, right now, the questions Dave was asking earlier, what causes this disease? What are the molecular and cellular drivers? Right now, nobody in the world, the smartest person in Castleman disease, does not know the answer to those. But we will work together, and as a group, we'll figure out the answers to those questions. When we do, we're not just going to change the lives of everyone here in this room, not just everyone around the world with Castleman disease, but everyone, for every future generation, they're going to know the answers to those questions because of what we, as a group, did together. The impact we're going to have, not just on the people in this room, but around the world and in the future, is going to be enormous. So many more patients in the future will have this disease. And all the work we're doing now isn't just for the people here, it's for them as well. We're really going to take this disease head on. We've got a great plan. We've got a great team. We're making progress on the stuff we've already proposed. We're moving ahead full steam. And the one thing I just want to emphasize again is, one project isn't enough. We need as many donations as possible, participate in the registry, get other people enthusiastic on social media about participating in the registry and about contributing to the biobank. We really need as many people in this as possible to get those answers to those questions so that we can change the future of Castleman disease for all of us. Thank you.
Jason, that was awesome. Thank you, Jason.
Thanks, Dave.
Jason, it's Grant. Just
Yeah.
You mentioned in a slide the research cycle that you
Yeah.
Is that different from other research projects you've been on, or is it a unique model?
Yeah, so that's actually pretty similar. So there are slight kind of variations on a theme where sometimes you might actually work back and forth between what happens in the patients, and before it's even an approved drug, you can look at what happens in the patients and go back to the research bench. But it's basically just that cycle. You have the patients, the physicians, the researchers get together. They create resources so you can do the research. They figure out the most important research projects. They get those done. As Dave mentioned earlier, normally the NIH gets that part done, they fund that. But unfortunately, they haven't been doing that for Castleman disease. So we've really taken that on as an organization. So for other diseases, it is a little different because they'll have more support from the NIH at that point, but we're pushing that ahead. And then once you get those good ideas, you start to bring those to patients. There are some nuances there I didn't get into. You have to make sure if you've got a good idea for a drug, you don't just put it in a patient right away. You have to test it to make sure it's very safe before you put it into a patient. So there are some regulatory things that I kind of didn't get into the details of. But more or less, it is that same cycle. So other people have done this same sort of thing before. We've seen it work before, and that's why we're so confident that it's going to work for Castleman disease. Any other questions?
Sorry.
No, please.
David's mentioned earlier on that you came on board. Why did you personally come on board to be part of finding a cure?
That's a great question. So I was actually a student at Penn Med with Dave. And I think Facebook gave me one of those reminders the other day. You know, on Facebook, it'll be like, there's this photo from so many years ago. Do you want to post it? And it's all this stuff coming up from from your past. I think it was one of Dave. It was in the first year, a Christmas party. Me, him, and a few other friends, kind of, arms over each other's shoulder, smiling into the camera and thinking, we were all good friends before this happened. And then when Dave got ill, the entire class rallied around him. Kind of a funny story. I felt there was this event that everyone went to. I didn't go to the event, so I felt really bad about it. So I sent Dave this long email to try to make up for it, explaining what I thought they could do for research and stuff. And Dave was like, hey, these are actually some interesting research ideas. We should think about this some more. And at that point, it became the sort of thing where it's very clear that Castleman disease is similar to other types of diseases that we're actually getting really good at treating. Like right now, we're much better at treating cancer than we were 10 years ago. Autoimmune diseases, which Castleman is also like, we're much better at treating autoimmune diseases than we were 10 years ago. These are diseases we know how to understand, and we know how to create treatments for them if there isn't already a treatment out there we can use. So as Dave has mentioned many times before, it's not a matter of everyone has been studying this for so long and no one's been able to figure it out. It's really that the tools were all there, a really interesting question that would really impact the lives of many, many patients and help a lot of people was there. You just had to bring those together with the right resources and the right people. And I think that's actually an exciting opportunity because getting to what I was saying earlier, we really are a part of history coming together as a group to say, we're gonna change this disease, not just for us, but for the future. And that's such an exciting thing to be a part of. So, yeah.
Of course, this goes for everyone at CDCN. Jason's a volunteer. This is like his time that he could be doing a million other things. This is what he does, and he puts his heart into it. I mean, it's amazing. Yeah.
Thank you, everyone.